In this video, we're going to talk a little bit about infectious disease, kind of do an intro to the big picture of how our immune system works, or at least rather how some parts of our immune system work. So what we're going to talk about in this video is infectious disease and how we fight and how we fight it off. So there are a couple aspects of it that we should think about. Is first, what is it? How do we avoid it? And then what happens in our bodies if we do become infected by pathogens, so that we can remain healthy and not die from those infections. So really, when we talk about infectious disease, what we should be mostly concerned with, first off, are pathogens. Pathogens. And pathogens are any living organism or virus that causes disease. So this is a definition that you need to commit to memory. Pathogens cause disease. These are living things or viruses that cause disease. So you should also note that based on this definition, we don't consider viruses to be alive. So the living things are the bacteria, the protists, the fungi, plants or animals, but plants and animals aren't considered pathogens. We just don't have those as pathogens. But those are the kingdoms of living things that you need to know, and viruses are not one of them, but viruses do cause disease. And we're going to talk about HIV at the end as an example of what's going on. But for some of these pathogens, we have these things that you're familiar with that we call antibiotics. So when you go to the doctor and they've determined that you've got, let's say, a bacterial infection, so this is most commonly used for bacterial infections. You will go to the doctor and you're sick, and they do a test and they determine that you've got a bacterial infection. One way they're doing that is by looking at your immune system response. So we're going to talk about white blood cells, and when you have high white blood cell counts, because your white blood cells are reproducing to create a chemical to fight off these bacterial infections, they know that you have a bacterial infection, and they give you antibiotics. And these antibiotics are targeted for these bacterial cells. Now, bacterial cells are prokaryotic. So we haven't talked about what that word means yet, but I hope that you remember that from 9th grade biology as well as middle school biology. Bacterial cells are prokaryotic, whereas our cells are eukaryotic. I'll spell it out for you. And there are some key differences between these. So these antibiotics target special features of the prokaryotes. For example, some might target their ribosomes. Ribosomes are cell organelles that make protein. So if we take an antibiotic that's designed to attack the prokaryotic ribosomes, just a foreshadow, those are the 70S ribosomes. They're going to target those ribosomes, not the eukaryotic ones, which are considered to be 80S. There's some key differences we'll talk about later. Because it's a different structure, that chemical will target it a little differently. And then what we have is other antibiotics that target cell walls. So bacteria have cell walls. They're not made out of cellulose, um, like plant cell walls are. They're made out of. They can be made out of another chemical called peptidoglycan, but these cell walls can be targeted. So it prevents. This antibiotic will prevent cells from creating new cell walls, which actually prevents them from growing in size and dividing into new cells. So that really does control the infection or the mass number of those bacterial cells. So it's kind of interesting that because we know there are key differences between prokaryotes and eukaryotes, we can design antibiotics. But what we should mention is that there could be some antibiotic resistance. And that's something that we had learned about when we talked about evolution that could develop from these antibiotics. So, so some bacteria that are targeted could have some genetic immunity. So they're not being targeted by this chemical, and it's genetic, so it's in their DNA, so they survive and pass down the genes, so the new population is immune. And that's not a good scenario. So you've heard of penicillin, I'm sure. The newer drugs are vancomycin or methicillin, and these are other kinds of antibiotics that are common now, but we're always playing this arms race. We're trying to kill these bacteria. Now when we kill some bacteria, we actually end up killing many more bacteria. So oftentimes when you're on antibiotics, you may have, I don't know if you have these or not, I hope that you don't, but you might have digestive problems, like diarrhea, or your stomach is hurting, you might be throwing up. And these digestive problems are because those antibiotics have also killed the flora, which is just, these are the community, this is the community of bacteria that live in your intestines. And some of them are good. Remember, some of them make vitamin K for our bodies. There's E. coli, good E. coli in our bodies. But it kills that flora, and then it messes with your natural digestive system. So you have some issues, but the goal is that you're killing some of the bacteria that might cause you disease. So there are pluses and minuses to that information. But I guess one thing that we should talk about is that these are not, antibiotics don't work on viruses. So I'm going to write that. Antibiotics don't work on viruses. And that's really because of the way that viruses work in our body. One way to remember that is anti means against, bio means life. Viruses aren't even alive, so antibiotics are not going to work on something that's not alive. But what's really going on is that these viruses use the host cells to reproduce. So they're not big enough to have their own ribosomes, because they can't make their own proteins. And proteins are very, very important for all living things because they regulate what goes in and out across the cell membrane, and we'll talk more about that in a future unit. But they use host cells to reproduce. So if we're talking about a human, these viruses inject, they're just a protein coat on the outside and DNA or RNA on the inside of them. And they inject that genetic material into the host cell, so let's say our cells, a specific type of cell in the human, and that genetic material finds its way into our own DNA in the nucleus. And it finds its way and it splices its way in, and so when your body is making its own copies of DNA, it unknowingly makes extra copies of the virus. That DNA gets converted into mRNA. Again, unknowingly, you're making viral mRNA. That RNA finds its way to the ribosomes to make viral proteins, and you're making hundreds of these new viruses inside a single cell, and the cells usually burst open, releasing these viruses, so these viruses then can attack more of your cells, and they reproduce in that way. But because these viruses use our cells, we can't. So we would have to target not prokaryotic cells, but eukaryotic cells. And I think you can figure out the problem with that, because all of our cells are eukaryotic. It just didn't look right to me. Because all our cells are eukaryotic, we would be attacking our own cells. That's not a good situation. We would be harming our own cells. So antibiotics don't work for that, because antibiotics are targeted for prokaryotic cells. So really the next question is, how do we prevent or avoid getting infectious disease? That's a great question. So we talk a lot about this in school. We teach you, when you're younger, how to avoid this by washing your hands when you're sick, and not sharing food or drink, especially when you're sick. But you could be in latency, so you could be infected but not show the signs of it and still pass on. So that's a danger. But really, the best answer is, how do we prevent this? We try to quarantine ourselves. So we avoid coming in contact with other people, especially when we are very, very sick. You don't come to school, partly because you're not feeling well, but partly because in our society, we don't want to spread that disease around. We want to avoid that as possible, but as you know, that's quite impossible. Again, you may not know you're sick yet and be passing it on, or you just can't afford not to be at school sometimes. So that's not the end of the answer, but that's one way that we can avoid spreading disease or contracting that disease. So really what we should talk about very quickly is what in our bodies help avoid us getting sick. So we're constantly being bombarded by pathogens, but we're not always getting sick because we have a couple of lines of defense. And the first is our skin. So there are a few layers of our skin, but the two that we're going to talk about very quickly are the dermis and the epidermis. A good way to remember this is that a dermatologist is a skin doctor, right? So the dermis are the living cells that are under the surface of your skin. This is where we have blood capillaries, we have nerve endings. The epidermis are the dead dermis cells that are pushed up or maybe pushed out to the edges. So your outer layers are dead cells, and because they're dead, pathogens have a hard time attacking them. And that's really why when you get a cut or an abrasion, you have to clean it, because you're trying to prevent pathogens from getting into your body. Okay? So I hope that makes sense to you. And it's really trying to find its way into your blood vessels, into your circulatory system, so it can move all around your body and try to target the cells that it needs to. We also have, as a second line of defense, stomach acid, but that stomach acid is really for waterborne and foodborne pathogens, because the water and the food are going to travel through your stomach. Right when you ingest the food, it goes in your mouth. It finds its way down your esophagus into your stomach, and that stomach acid, which is hydrochloric acid, which has a pH of about 1.5 to 3, so it's highly acidic. And remember, it's acidic so that the proteases, pepsin in particular, is at its optimal pH, so it's not denatured and it functions very quickly. But the other reason that it's highly acidic is that it helps prevent us from getting pathogens, because this acidity breaks down most pathogens. Most. Now you might have heard of food poisoning, particularly, I hope you've heard of E. coli 0157. This is a strain of E. coli. So I mentioned earlier we do have E. coli in our intestines, but this is a strain of pathogenic E. coli. It's bacterium. And this E. coli is really breeding in our cow populations. And it's in their intestines. So it comes out of their intestines in their manure, and when that manure finds its way either into your meat, because it's not necessarily the cleanest of environments. Those cows and bulls are in pens in which they're often wading, W-A-D-I-N-G, not waiting, around in their manure, and because all of the meat and muscle mass is mixed together, E. coli can spread very, very quickly into the meat, or if that manure becomes leachate and spreads to other crops, even things like peanuts and spinach, or could be used as a fertilizer and has this E. coli 0157, it can spread, and this is resistant to these low pHs or highly acidic environments. And really what we're doing is these are being bred in these cow populations because of the food that we're feeding them, because we're feeding them corn, which is subsidized, which means that it's cheaper for the market. It costs less because the farmers are spending less money on the corn, so they can sell the meat for cheaper, but then there's this higher health cost for treating someone who has this kind of food poisoning. Anyways, I just wanted to give you a little bit of context on that. And then the next thing is mucus. So this is for airborne pathogens. And this mucus lines passages or entrances into our body. For example, the trachea, which is not the same as the esophagus, but this is the other tube that's separated by the epiglottis that controls which tube is opened up to the atmosphere. So the trachea leads from your nasal cavities into your bronchi and then into the alveoli of your lungs. But this is lined in mucus, which, you know, like if you've ever coughed up mucus, it's from the trachea. We also have mucus in our nasal passage. You notice snot. We've also got mucus lining in our urethra, which is a tube that goes from your bladder to the outside of your body, and then in the vagina, which is really to protect the uterus and the ovaries from pathogens. And so some of these cells that make the mucus are lined with cilia, which are the short hair-like structures that you might have learned about in 9th grade. We often talk about them as things to help cells move, but these cells aren't moving. What they're doing is kind of they're pushing the mucus up and out of the body if it's filled with pathogens, and these cells that make the mucus make lots of lysozymes which are a type of enzyme found in lysosomes, which we'll talk about, but these are parts of cells that lyse, so think about hydrolysis, right? Lyse means to break. So these are enzymes that break things down, particularly pathogens. Okay, so so that's how we prevent disease or these pathogens from getting in. But that's quite a lot of information, but we're not quite done yet. So again, I'll tell you the immune system is kind of complex, but it's not too hard if you take a little bit of time to make sense of it. So so let's talk about this. What if pathogens get into your body? What if they get in? What happens? Well, we have a certain type of cell in our body, which we've talked about before, which are called leukocytes. These are white blood cells. And I think you've heard of these before. I'm going to abbreviate these as WBCs, but you would never get away with that on an exam, so I would not get in the habit of writing that. But there's several types of white blood cells, and oftentimes we teach kids that there's only white blood cells. There's several types of white blood cells. So the first one we'll talk about are called macrophages. So macro means large, and these are large cells that that are non-specific. So these are large white blood cells that are floating around in your bloodstream, and actually they can change shape. If you ever, sometimes it's called amoeboid movement. Basically, they can move around like an amoeba and squeeze between the cells of the lining of the blood vessels and find their way into other cells, or between other cells. But these are non-specific, and basically they're going around your bloodstream, constantly assessing other cells or chemicals to see, are they self or non-self. So they're reading the types of proteins by interacting with the proteins on the edge of the macrophage itself to recognize, are these proteins that are part of the DNA of this person, or are they not? And if they are part of themselves, then they leave it alone. And if they are not part of that person, or coded from the DNA of that person, then what happens is that there's phagocytosis. And that's really where the phage part comes in. So phagocytosis. So cyto refers to cell, and so what this really means is that the non-self cell is engulfed by the macrophage or macrophage. So it has to go inside of this large cell. So let's draw it as a large cell, and we've got a bacterial cell here, and basically what's going to happen is that it's going to go inside of the macrophage, and then it's going to be, so here we go. So then it's going to be inside the macrophage. And then there are going to be lysozymes that break down this cell. So, and then it spits out those parts out of the macrophage. So that's kind of, I think, kind of interesting. But these non-self proteins that are on the cell membrane of these other cells are referred to as antigens, and that kind of leads us into the next section here. So one type of leukocyte is the macrophage or macrophage, and then the other kind that you need to know are the B lymphocytes. And you got to keep this straight. This is a type of leukocyte, but it's a lymphocyte. They're found in your lymph nodes. Okay, so these B lymphocytes or beta lymphocytes are ones that make antibodies. Antibodies are proteins that your body makes to bind to antigens of a pathogen. Alright, they bind to the antigen of a pathogen. So remember that an antigen are the non-self proteins, and the macrophage is non-specific, but the B lymphocytes are specific. So we have lots of different kinds of B lymphocytes based on the different kinds of pathogens that have made it all the way into our bloodstream, past our skin, past the mucus, past the stomach, and the B lymphocytes are very specific for that. So what ends up happening is that when you get an infection from a pathogen, we have these cells that are called helper T cells. Helper T cells. And they're floating around through your blood. There's another type of white blood cell, or leukocytes. And they are recognizing self or non-self, kind of like the macrophage does. Except that when it recognizes something as a non-self, it assesses what kind of non-self cell it is. And then it sends a signal to the B lymphocytes. So it sends a signal to those B lymphocytes that are floating around. There aren't many of them because there isn't that much space because there's lots of other things happening inside the blood. And these B lymphocytes reproduce through mitosis, which we'll learn about. So they make lots and lots of copies of themselves, and then they trigger antibody production. So they start making lots and lots of protein antibodies so that they can then attach to the antigens that the helper T cells recognized and break down those pathogens. And then when there's a high response like that, where there's a lot of these B lymphocytes from mitosis, what lingers after this whole reaction happens are called memory cells. And they're floating around in your blood, not in high concentrations, but enough so that if you had a secondary infection of the same kind of pathogen, you already have antibodies available to attack. Alright, so that being said, let's talk a little bit about HIV, and then that'll be the end of this. So you should know what HIV stands for: human immunodeficiency virus, and this can cause AIDS, autoimmune deficiency syndrome. So it can cause that, but doesn't always cause that. And again, you need to know what these things stand for. But the HIV is human immunodeficiency virus. So it's a viral infection. And in fact, this is a retrovirus. So retro means backwards or old, right? So this retrovirus has RNA in it. That RNA goes through a process that we call reverse transcription. We'll learn more about transcription translation soon, but this is basically where it goes from RNA backwards to DNA. And then that DNA finds the DNA of helper T cells. So what's interesting here is that this virus is, it's got a protein code. It's not a cell membrane, and it's got proteins that stick up off of it that have these little signals on it. And this is a terrible drawing, but they've got these proteins that stick off of it, and they're floating around the body searching for something that it will match with. And in this case, the HIV matches with helper T cells. And so what the virus does, as I said before, will inject its RNA into the host cell, the helper T cells. It does a reverse transcription to make DNA. That DNA finds its way into the nucleus of the helper T cells. Helper T cells are making lots and lots of copies of that virus, and then the process of that kills the helper T cells. Now, if we go back and we see that we've got helper T cells here, if the helper T cells die, then the B lymphocytes never get signaled to reproduce, trigger antibodies, and attach those antigens to a new pathogen. So often what happens is, because those helper T cells are so low, someone with HIV or AIDS, and basically AIDS is when these helper T cells hit such a low volume or low concentration in the body that basically the person with this can't fight off a secondary infection. So the cold, the common cold, or the flu, or any other kind of reaction that would normally be able to be dealt with won't be fought off appropriately because these helper T cells have been killed. And I hope that makes sense to you. So there are a couple of issues. I'm not going to write these, but I'm going to just talk through these. A couple of issues that are related to HIV and AIDS. Obviously, one of them is, because this is viral, it's very difficult to come up with a vaccine, because we would be targeting our own cells to try to get rid of this vaccine. There are antiviral medications, but HIV is really good at mutating very quickly, so those antivirals don't work 100%, because there are always going to be some viruses that are different, even within one person, that manage to avoid being destroyed by these chemicals. There's also been a social stigma associated with HIV. It was associated with homosexuals and drug users, and so because of those associations, although those are not the only populations that were affected or are affected currently, so but that's the stigma that it had. So it was very difficult, especially in the 80s, to get much research or much money for research on HIV, although times are different now and there's a little bit more money available. And also, so besides drug users who were getting HIV through shared needle use, there was also the potential for people to contract HIV through blood transfusions. So I was born in 1984, right before the federal government mandated that blood in blood banks be tested for HIV. So that's actually very scary to think about, because so my mom had a C-section with me, and she could have contracted HIV. There's a possibility that I could have as well, because of the blood transfusions that were necessary because of the loss of blood during a surgery like that. So it's kind of scary to think about, and that was in 1984, even though they knew that this was happening in 1981. Because of the social stigma, the federal government did not recognize HIV or AIDS for another couple of years before. And when they finally did, they were able to make mandates about testing. But anyways, I think that's the end of this video. I know it's a lot. I hope that it makes sense to you. And if not, jot down questions so we can talk about them in class.